Now, this next bit is an article from here. And I kid you not, this is from the Institute of Virology in Wuhan. This is where this research on monkeypox is being carried out. So let's look at this. Let's look at this in a little more detail. Now, this is an article in Virologica Sinica, which we'll look at in a minute. That's the article there. There it is. Download the PDF. It is a full peer-reviewed article in this official Chinese publication. Now, let's look at what they're doing. What they're doing basically is they are working, and this was published in February 2022. So the Wuhan Institute of Virology is working on a test to test for the presence of monkeypox. And I think we could assume they're interested in tests for smallpox as well, bearing in mind this is a potential biological warfare agent. If you're already starting to feel uncomfortable, join the club. I'm actually very uncomfortable about what I'm about to tell you. So that's the name of the paper there. Efficiency of a large-scale fragment of monkeypox virus. So basically what they're doing is they make a section of monkeypox virus. And they want to use that as a quantitative polymerase chain reaction template, and they're going to use this way to put it together into a yeast. Now, direct quote from this paper. Since monkeypox infection has never been associated with an outbreak in China, the viral genome material required for PCR detection is unavailable. So the Chinese authorities, it would appear, are incapable of getting hold of monkeypox virus. So they have to basically make their own using synthetic techniques, or make a part of the viral genome themselves using synthetic techniques. So the monkeypox virus is available readily in labs in Nigeria, Congo, the United Kingdom, presumably now in the United States, several places in Europe. And the virus is available from anyone who has symptomatic monkeypox. Anyone who's got a pustule with monkeypox, just take a scraping and there you've got the virus. So the idea that the Chinese authorities can't get a sample of the actual virus and have to synthesize their own, I'm not buying it. I'm not buying it. All they need to do is send some people to Nigeria, slip the Nigerians a few dollars, and I'm sure they'd come back with a vial of monkeypox virus. But apparently they can't do that. Why on earth would they publish that? It is so obviously incredulous, saying that it's not available in China, so they've got to make their own. Don't buy it. Do not buy that at all. You can make your own mind up, of course, but I'm giving that a red cross. I'm not buying that one, I'm afraid. It's available. The virus is available, especially to somewhere like the Wuhan Institute of Virology. So anyway, because it's not available, they're forced to make their own, poor things, using viral DNA recombinations. And this technique here, transformed associated recombination, where they put the part of the viral genome together to assemble large DNA constructs. So they're actually assembling. So obviously they know the gene sequence in the monkeypox virus. So they are synthesizing that basically from scratch, putting the DNA bases together, and then they put it into a yeast to multiply it up so you can make any amount of it, and then you can use that yeast to test the veracity of your quantitative polymerase chain reaction tests for monkeypox. That is what they are suggesting here. And then made of 55,000, so it's 55,000, 55,000 bits of the monkeypox viral genome, and then they put it into this. Now this VL648B is a standard commercially available yeast cells. So basically it means you can brew it up like beer once it's in yeast cells. Once you've got yeast cells, you just basically put them in water with a bit of sugar and they'll brew up as many as you want. Basically that's the case. So you can make up huge amounts of it. Now, what is Virologica Sinica? Here it is. Now, this is the information about that. It is readily available. And I'll tell you a little bit about what it is from this website. It's the official journal of the Chinese Society of Microbiology, and it will serve as a platform for the communication and exchange of academic information and ideas in an international context. Sounds pretty good. Now, let me just show you something here. This is direct from this paper, because this is the bit that's really quite chilling. Here it is. This work was carried out, just in case you think I'm making this up, at the Wuhan Institute of Virology. This is one of the reasons that it's so uncomfortable, what I'm telling you. Stroke terrifying. Discussion section of this paper. So actually in this paper itself, it says this: however, this DNA assembly tool, putting together the viral DNA, remember this is the instruction to make the viral proteins, the monkeypox. And by the way, if you could make a monkeypox virus, I'm sure you can make any other viruses you want. For example, just think of a random one, smallpox. But smallpox is already saved already. We know that the original strains of smallpox are saved already. But whether they're saved already or not really doesn't matter anymore, because they can now be synthesized. Because the genome is known, the whole virus could be synthesized. Not saying they're synthesizing the whole smallpox virus, just saying it could be done. And that doesn't apply just to China anyway. That applies to everywhere that does virological research. Right. They say this DNA assembly tool applied in virological research could raise a potential, I'm going to start using a red pen here, this is pretty alarming stuff really, could also raise potential security concerns. Well, imagine that. I should think it could. Especially when the assembled product contains a full set of genetic material that can be recovered into a contagious pathogen. So the genetic material can be taken from the yeast, put back into a virus, a contagious pathogen. Recently, a group of scientists was funded by a biotech company to synthesize a full-length horsepox virus genome and recovered it into an infectious virus. So this has been done. In other words, it can be reverse engineered. Can be reverse engineered from the yeast back into an infectious virus. Now, I've left all the hyperlinks here, so you click on these in the descriptions, it'll take you straight to them. Not surprisingly, such a controversial treatment has received enormous attention and raised global debate on its biosecurity implications. Again, hyperlinks there. So it seems the Wuhan Institute of Virology is very aware of these risks and is talking about it. Of course, this paper has been approved by the Wuhan Institute of Virology, which, as we've seen, is not open to international visitation, shall we say, let alone international scrutiny. Again, not just the Chinese situation. This would be the same in Portugal. It would be the same in the American facilities where potential military threats are addressed. But some might say that the Wuhan Institute of Virology has been leaky in the past. We have no definitive proof of that, but that has been suggested. But fortunately, the Wuhan Institute of Virology go on to say, in this study, although a full-length viral genome would be ideal reference template, so it would be much better if they could make the whole thing, we only sought to assemble a 55 kilobyte viral fragment, less than 1/3 of the whole monkeypox genome. So in this publication, what they are saying in this publication is they've only synthesized 1/3 of the genome. If they have synthesized the other 2/3 of the genome, they're not writing about it here. And of course, we've no reason to suspect that because it's not written here. They've only synthesized 1/3 of it. it would have been better to synthesize the full thing, but because they're responsible and being careful, they haven't done that. At least they haven't written about that in this publication. This assembly product is fail-safe by virtually eliminating any risk of recovery into infectious virus while promoting a multiple target. So basically they can use this for testing, but it's perfectly safe because it's only a third of the virus, therefore you can't recover the whole viral genome. Of course, it would also mean that any less ethical viral constructor in other parts of the world would only have to bother constructing 2/3 of the virus rather than the whole thing. There you go. I think I've made my concerns about that clear. And more references there. Now, just to quickly recap, this seemed to have come from Nigeria, this strain. It seemed to arise in male homosexual events in Spain and Belgium. Gay events in Spain and Belgium, and spread from there. But the UK seems to have got the most number of cases. So that doesn't quite add up. Maybe there's something we don't quite know there yet. But this is spreading in male homosexual communities. Now, I don't see any particular reason why that should be so. I suspect close proximity in heterosexual sexual activity, or not even heterosexual activity, just any close amorous activity would be sufficient to spread the virus. I suspect that the fact that it's spreading in the male homosexual communities in Europe is a founder effect. It's just where it got to first. And of course it can spread to other people, certainly because it's a transmissible disease. Unless there's more to come on that, there may well be more to come on that. But in the UK at the moment, last 24 hours, another 11 cases, now got 190. We've ordered 20,000 additional doses of smallpox vaccine. Note, a smallpox vaccine, not a monkeypox vaccine, a smallpox vaccine. Offered to close contacts of those diagnosed with monkeypox to reduce the risk of symptomatic infection and severe illness. So the smallpox vaccine offering protection against monkeypox. Just the same as back in the 1790s, Edward Jenner's cowpox pus from Sarah Nelms, the cowpox that he took the pus from, protected James Phipps, the young boy who he inoculated with cowpox to protect against smallpox. So in the same way now, the smallpox vaccine is giving this cross immunity to protect against monkeypox. Dr. Rosalind Lewis, WHO expert, it's very important to describe this because it appears an increasing mode of transmission that is been unrecognised. She's talking there about spread in the sexual context, particularly in male homosexuality context at the moment. Unfortunately, it will be unfortunate, she says, if monkeypox were to exploit the immunity gap, strange term, I'm not quite sure how scientific that is, left by smallpox 40 years ago. So it's as if there's kind of an it seems to be saying that she's saying that there's kind of an ecological niche, a place to live, that smallpox used to live. But a similar virus could kind of take over that niche, which is a bit strange, really, because smallpox has been eradicated, eliminated. So don't quite know what her thinking is there. I'm not quite sure how scientific that is. It's as if each disease has got its own little niche, and if you take that away, another disease will come along to fill it. I'm not sure there's too much scientific evidence for that. But anyway, that's what she's saying. Anyway, I agree with her. That would indeed be unfortunate. This is not a disease we want to become endemic. And she says there's still a window to close the outbreak. Let's hope that that happens before it gets into animal reservoirs. Because if it gets into animal reservoirs, it's going to be very hard to get out of animal reservoirs. So there you go. National Institutes of Health and Wuhan Institute of Virology both seem to have uncannily anticipated this. Make of that what you will. Thank you for watching.